I'll be talking to you about this correlative cryo-SEM and cryo-NanoSIMS imaging technique. And as mentioned by Christelle, I'm working in the lab of Professor Anders Meibom at the Laboratory of Biological Geochemistry at the EPFL. So to give you an outline of the talk for today and what you're going to be hearing. So first, I thought I would introduce the topic of NanoSIMS and what is the NanoSIMS as a technique. And then what are the applications we have so far where NanoSIMS has been used in biology, and the challenges that we face in the application of this technique, the way it is for biology, and how this has been overcome now with the help of a cryo-NanoSIMS. And I will talk about the workflow that's been developed for the correlative imaging, and use salt stress in plants as an example to show a real application of the cryo-NanoSIMS imaging technique. So I think we have many imaging enthusiasts in the audience here. So I thought first I would place NanoSIMS imaging itself in the broad spectrum of imaging techniques that we have. I think many of these techniques must be familiar to some of you here. And so when you think about a biological question, NanoSIMS, for example, is an isotopic and elemental imaging technique, which works with a lateral resolution of about 100 nanometers up to about 300 nanometers. So in terms of biology, if you're interested in looking at things at the subcellular level and what's happening in the organelles, this would be one of the techniques that you would go with, and especially to map the elemental and isotopic compositions in there. And so what is NanoSIMS? So NanoSIMS is a technique that is based on the principle of secondary ion mass spectrometry. So what you have is a very finely focused primary ion beam that is charged, which is bombarded on the surface of a sample. And this then affects the top few atomic layers of the sample in a process called sputtering. And it causes releasing of different ionic species. And from these ionic species, you basically extract out all the secondary ions and then run them through a mass spec analyzer, and then you separate them based on their mass to charge ratio. So here is a schematic of a classical NanoSIMS instrument. And if your sample sits in this position here, you have a primary ion beam that is being generated from the ion source, which is then incident on the surface of your sample. And this finely focused beam is then extracted out using a range of ion optics, and then passed through a really large magnetic field. And within this magnetic field, you basically deflect the ions based on their mass to charge ratio. So the lighter ions get deflected more, and then the heavier ions get deflected less. And so you can then place 7 different detectors in positions of the masses that you're interested to analyze. And then you can start to generate, and you can start to run the beam, raster the beam across the surface of the sample and start to generate a chemical map of the sample that you're interested to image. So here, for example, you can look at 7 different ionic species that have been captured with the 7 detectors, and they all correspond to the exact same position on the sample, and they're being mapped in parallel. And so that is one of the biggest strengths of this technique. And to give you a feel for what's the size of this image, you usually classically a typical NanoSIMS image is about 256 by 256 pixels, and typically about 30 to 50 microns in size. And so with NanoSIMS, what you have is the strength of really good, very high spatial resolution, which is really important for many of the biological questions. And so you really work with a resolution of about 100 nanometers to start with, and then you can really expand the applications there in biology. And so then you also have the strength of having very good, very high mass resolution. And so this is also another very powerful feature of the NanoSIMS. And as I mentioned before, so you can really detect 7 different elements in parallel. And so this is really useful when you have biological questions where you're trying to understand how one element is kind of moving, functioning in parallel with the other, on how when you affect one of the ions that you're interested in, how does this actually reflect biologically with more than one ions? And this is really powerful for biology again. And it has a range of imaging applications. But one of the interesting things I wanted to highlight was it's also very useful when you want to track isotope uptake and distribution within a biological system. So, and then you look at its incorporation at like the subcellular level over time. So this is really another very important function use of NanoSIM. So I thought I would take two examples to kind of show you its application in biology. So here, for example, I talk about one of the examples where they were using NanoSIMS to look at DNA synthesis. So they basically used 15N labeled, thymidine labeled nucleotide base, and they were interested to see how the chromosomes segregate in the DNA. And so on the left, you have an image of phosphorus, which reflects the chromosomes within the nucleus. And what they did was they looked at the incorporation of this 15N labeled thymidine in adult mice in newly dividing cells. And so they wanted to see if the chromosomes are segregating evenly or unequally, and if the newly dividing cells actually incorporate this DNA, or and many other different applications that it can be used for. And this is useful because then in medical biology, for example, they're interested to understand if disease models have defects in their divisions, and this can be one of the techniques which can really help in that kind of, for that kind of biological question. And then another example I wanted to highlight was its use in looking at cerebral glucose metabolism. And here in this case, what they did was they fed the mice with 13C labeled glucose, and they wanted to see where this glucose that's being fed to the mice ends up in the brain. And so here they were interested in looking at different types of brain cells. And on the left here, you can see an image, which is a TEM image of the brain cortex. And you can see here is the nucleus of the cell. And in this TEM slice, you can actually get an overview of what is the structure. And then you can see where the 13C labeled carbon isotope has kind of ended up within the brain tissue. And in this case, it's kind of, you can see that it's strongly localized to the nucleolus, and also in Golgi bodies. And so so there's many so this is really interesting again to see how the destination of a compound is within a tissue, but it is really important to also remember that you can say a lot about the location of the isotope in the tissue, but you don't get any molecular information as to which compound this isotope is actually is present within this material. And if you look at any classical sample prep technique for NanoSIMS, it involves the classical EM sample prep. And I don't know how many, if some people may be familiar with how a sample is prepared for EM, but one of the classical first step is to basically fix a tissue, and then you run it through a series of dehydration steps by which you basically remove all of the solutes and all the soluble compounds from the cell. And so what you're left with is things that are actually incorporated into the fixed structures, and that is what you can actually measure here. And so this is really important for us because in biology, because this can answer already a lot of questions on things that are actually fixed within the tissue. But a lot of biological tissues have compounds which are very important in the solutes, for example, ions. And these are usually drained out of the cell when you actually run it through a classical sample prep. And so what we needed was, if we wanted to, and this is one of the biggest caveats of using NanoSIMS for biology. And so if you really want to visualize the distribution of the soluble compounds in the cell and see where they are and what their function is, you would need an instrument that can actually work under cryo. Because currently cryopreservation is the best way you can actually keep all the soluble components in the cell intact and be able to look at it. And so you need an instrument that can work under cryo. And this was what was done by Professor Anders Meibom here and his team. And so they basically took a conventional room temperature NanoSIMS and made it completely cryocompatible. And so this instrument can now hold a sample which is vitrified and preserved under cryo temperatures. And then now you can really start answering some of these questions in biology, which were hard to answer before. And you can see the strength this has for the biology. And I will talk more about this in the coming slides. And so here comes my plant biologist geek in me, where we were in for a long time. There is this question of salinity stress, which I wanted to introduce to you. So salinity stress is a massive problem for global food production. And when I say salinity stress, it is mainly related to the ion of sodium. So for us as humans, we really love sodium. We like sodium salt in our food. Our cells like it because they need it for different signaling aspects. But a plant is not really a big fan of sodium. And most of the cells actively try to keep this ion away. And we understand that salt is a problem for the plant, but we don't really understand what exactly is happening within the plant. And this is what is a question, and this is a problem that is really been difficult to answer for a long time. So what we have is a problem of scale and spatial detail. So we can look at field trials and see what happens as a consequence of salt stress to the plants. We can look in the lab and try to start understanding what is happening to the anatomy of the plant and how salt is actually impacting the plant growth and development. But what we could not still answer is, how is the sodium actually sitting within a cell? And based on where it sits, what kind of processes is it impacting? And how does this actually reflect in terms of like toxicity within the plant? And so cryo-nanosims actually is now one of the ways by which we have actually extended the analytical capacity to be able to look into a cell and start answering some of these questions. And this is also very interesting because we have a lot of understanding about transporters, for example, which are broadly important for the plant to transport these nutrients. But what we don't really know is that within a cell, how is the sodium compartmentalized within the cell, and how do the plants actually respond at the subcellular level and redistribute these ions when they have a salt stress? So this is a question we kind of wanted to take on. And as Christelle mentioned as well, so this is the work I'm doing with the lab of Professor Nico Gellner, who's also here, and also with Christelle. And we are trying to look to see if we can answer this question of salinity stress a bit more in detail using the cryo-nanosims. And so the experiment. So I thought I would use this as a way to kind of take the example that I am working with to kind of walk you through the whole cryo-nanosims workflow. And so the first step before you do before you start using something is to actually look at how you would set up your experiment. And so in the case of plants, we have this plant which we call Arabidopsis thaliana, and this is our lab rat for the people who don't know. And so we have a wealth of genetic resources and a lot of different capacities to use this in the lab to understand what's happening in the plant. And so we use this as our model system, and we can subject this plant to different range of salt stresses and then see what is happening within the plant. And we also have genetic mutants for some of these transporters that have been suggested to be important for transporting these ions. So we can now play with these mutants and then start to see what exactly is happening at the subcellular scale using the cryo-nanosims. And so here I give you an overview of the cryogenic workflow for the correlative SEM-CryoNano and the cryo-nanosims. So it is a lot of steps. And so cryo-nanosims is like the final destination that we need to get to be able to make these maps. But you need to have a whole series of workflow in place to be able to actually take your sample and then keep it under cryo temperatures and then be able to image it in the cryo-nanosims. And so I'll walk you through some of the key steps a bit more in detail. And so once you have, like I said, we have in my case, I look at plants. So I'm stressing them with some salt stress and then starting to see what is happening. And so once I have a biological tissue ready, the first step is to basically high pressure freeze the sample. So why is high pressure freezing important? Because as I said before, we need to be able to maintain the sample in its most pristine state. And the best technology so far to keep a sample in this state is by high pressure freezing. So some of you might be familiar with different types of cryopreservation techniques where you can drop something in liquid nitrogen and then start looking to see what is happening inside. But here, it is important to note that the most important critical step is to prevent formation of ice within your sample. So when you have a living cell, which is kind of like in the case of plants, it's like 80% water, you don't want this to just be dropped into liquid nitrogen because it basically causes ice crystals to form and destroy the subcellular structure. So we need to use high pressure freezing because when you high pressure freeze, you basically take a sample and drop it down to liquid nitrogen temperatures, but you do this under very high pressure. So you go to, and within very few milliseconds. And so what this does is that it basically takes the water in the cell and instead of getting crystallized, it just converts it into an amorphous ice. And so now you have your sample in a really nice, clean, glass-like state. So all of your cell contents are well preserved. You keep your cell structure, and now you can actually start looking at the sample a bit more in its best state. And so after we high pressure freeze the sample, in this case, it is a plant root. You can see this like about 2 millimeter in size. This is something you really need to note if you were thinking of using this, because you really need to be able to fit your sample into this little planchet. And once you go through the high pressure freezing, all of the steps from here onwards are done under liquid nitrogen temperatures. So you have to really maintain the sample in this cryo state and then walk it through the entire workflow. So the next step is to plane the sample. Because NanoSIMS is a very surface sensitive technique, you need to have a really flat surface and to be able to actually image this sample surface to be able to image it. And so we use cryo ultramicrotome and with a diamond knife and plane the surface of the sample to create a really nice, neat, flat surface. And in this process, there can be a lot of contamination still with ice dropping on the surface of the sample. And this interferes with our imaging. And so it's important to then move the sample to etch the sample to get rid of some of this contamination. And so we use what is called VCT, which is a vacuum cryo transfer. And so this is like a little shuttle, which is under liquid nitrogen temperature, and you can keep your sample well preserved under vacuum, and you can move it through different instruments. And so it makes it compatible across all the different instruments in the workflow. And so when you freeze etch the sample, what you do is you get rid of some of the ice, you sublimate the ice that is on the surface of the sample, and you also introduce mild topography, because this then becomes very useful when you look at it on an SEM to start seeing the subcellular structures. And so then you coat it with a little bit of metal and take it onto an SEM, and then you start imaging the ultrastructure. And then once you have imaged it on the SEM, the same sample is then transferred with a VCT, coated with a little more metal, and taken to a cryo-NanoSIMS. And in the cryo-NanoSIMS, you can now start mapping it for its elemental distribution. And so the cryo-SEM that we use is a Gemini 500, which has a cryo stage, and it's with a cryo transfer system. And this is run by Christina, who is the SEM lab manager for this. And so we use this instrument to now take our root tip and start mapping the surface. So I show you now one of the first images of the Arabidopsis root tip in its most pristine state. So this, I hope you can appreciate, is a root tip that has not lost any of its compounds. So you really have not put any stain on the sample, and you're looking at a neat slice right through the root and looking at the different subcellular structures. So you can start to see all of these little dots that you see are nuclei, for example, and these little whitish looking structures are vacuoles. You really have a well-preserved root now with all its constituents nicely maintained in place. And you can now start kind of probing this to understand what's going on in different parts. And as a plant biologist, I can tell you which cell type is what and what function it has, and what kind of things that you can expect biologically to study from these different cell types. And so here are some more pictures. It's an imaging seminar. I'm not going to go short on pictures. So here you have a cell which is being imaged a bit more close up with the SEM. So you can start to see the nucleus and the subcellular structures and some of the other vesicles and mitochondria, for example. And here you can see a vacuole. You can spot the cell wall here, which is quite a thick structure here. But actually, it's a few nanometers in size. And also, I'm going to talk mainly about plants, but I also wanted to say that this is really any material that can be vitrified and is compatible with vitrification can actually be imaged on an SEM. And so here, for example, is a C. elegans for people who work on who are interested in worms, or you have some cells, fibroblasts, and you also can look at algal symbiont, which is something which many people in the lab work with. So you can really think about many different questions with the help of this. And so coming back to roots, because this is where I'm going to be talking about mostly. So in an Arabidopsis root tip, once we have a picture like this, like I said, I can tell you where we are within the root and what we are interested to understand. And so I can now use this as my reference to then mark areas that I'm interested to image further. And here, for example, I show you the image, the size of an image, which is a typical NanoSIMS image that you can get on this tissue. And I mentally mark all of these regions that I'm interested to then take it onto the NanoSIMS and then map with the cryo-NanoSIMS. And so here I give you one of the first images that we got. So on the left, you have a block phase SEM image where you can start to appreciate the cell ultrastructure. So you have the nucleus, you have the vacuole, and all the different subcellular structures. On the right here, I present to you a cryo-NanoSIMS image which is being mapped with the cryo-NanoSIMS. And here I show sodium distribution within the cells, for example. And the neat thing with this is that now we can actually go overlay these two information over each other, and we can start to say start to answer questions as to what is it that is actually accumulating in us in a different in a vacuole, for example. What is accumulating in the cell wall? So in this picture, it's telling us that the sodium is sitting in the cell wall. What does that mean for biology? We can start to answer all these different questions. And like I said, it's not just one ion that we look at, but we can look at like 7 different ions in parallel. So for example, in the same tissue, in the same position, we can look at sodium, and I have a picture of potassium, calcium, magnesium, sulfur, manganese, iron. So these are all some of the physiologically important ions for the plant that I've picked. And we are looking at the same exact location, and we can now start to see how these ions are actually redistributing within these tissues. And it can really help us it can already tell us some interesting information. Like, for example, we can see, like I said, you can see that the sodium is kind of sitting outside the cell and it's accumulating in the wall. The potassium is nicely distributed within the cell, which fits with the biology, because you know that potassium is important for many of the cellular processes. And you can see the calcium is bound to the cell wall. And there are many interesting observations that can be made. There's like a wealth of information here, but you're kind of left with more questions than answers at this point. And then you can really start to think, what does it mean when you have an ion in this subcellular structure? So what is it who is it working with? What are the compounds they are bound with? How are they redistributed when you give it a stress? And so this is what gets interesting now. And so this is what we did. But before that, we also tried to, because we use the cryo-SEM, which is one of the best cryo-SEMs we have. We also look to see if there are other e-beam techniques which have been, which is with the cryo-SEM. We look to see if we can actually look at the same ion distribution with this, but we couldn't really get the same sensitivity and resolution that we get with the NanoSIMS. And so we really have one of the best ways to actually image a cryopreserved sample now with the NanoSIMS. And so, like I said, so now that we start to kind of get a picture of what it looks like within a cell, so then the question was, now we can start to play with the salt stress. And so we started by giving it a very small amount of salt, which is not toxic to the plant, and it can kind of cause and start to see where does this sodium actually go into the cell, and where does it actually go and sit. And so from this picture, we can already see that the sodium nicely sits in the cell wall. So it's being kept away from the main core of the cell, and it's kind of excluded from the cell, and it's sitting in the cell wall. And the potassium is nicely distributed within the cell in all the subcellular compartments. And so this is really telling us that at this stage, with this mild stress, the cell doesn't want to take this ion in, and it's keeping it out. And so that is very interesting. So the next step is we then push the salt stress to a little bit more, but not toxic yet. And then we start to see, when we push the salt stress, what does the plant look like? What does the sodium within the cell look like? And so here is a picture of a plant which is subjected to a moderate stress. And what I hope you can see that these structures, which we call vacuole, which are within the cell, which are like the water-heavy structures. And this is where the ions are supposed to be kind of accumulating when they are in excess. You see that the vacuole is strongly taking up all of these excess ions, and it kind of really neatly, with the picture, shows us that when the plant has too much of these ions, which it does not like, it starts to transport it actively into these little vacuolar structures. And then you start to see that these ions are now transported in there. And so this really, with one picture, you can really start to capture a lot of information which we did not know before. We had a lot of inferences by staining with different chemicals. We had proxies of what a plant does when you actually put a lot of salt stress. You can probe an electrode and start to see what happens. But to actually look within a cell, and by putting a stress and to see how this ion is moving within a cell, or like redistributed within a cell upon a stress, was not possible. And we can now actually really get a lot of information with this. And so, like I said, we have, like I said before, we have quite some understanding of some of the transporters which are important for this, for the transport of this ion. And the first thing to do is, once we see that these ions are now being accumulated into vacuole when they are under the stress, if we take away one of the transporter that is important to move this ion, what happens to this sodium? Does it still actually accumulate as we expect it to, or does it change? Or we can start to understand the function of this transporter within the plant with this. And so we took a transporter which is important to transport this ion out of these out of the cell out of the cell, and then we subjected it to the same cryo workflow. And here, I hope you can see, so this is a mutant which was looked at, and these are the same subcellular structures which I call the vacuole. And I hope you can see that these structures now, even though the cells are under stress and they are bombarded with quite a bit of salt, they are not putting the sodium into these subcellular structures. So this really gives us a very nice visual information on the function of this mutant, which we did not know before. And actually, there's a whole story that could go on behind it, but I would not bore you with the details. We can talk more about it if you want. But this shows how powerful it is to be able to actually visualize this difference within the cell and how these ions are redistributed within the cell using the cryo-nanosims. And then, of course, we can't stop with this. And so we want to really push the stress a little more and see what really happens now to the plant. And so for a wild type or a normal plant, which has all its components intact, you see that the increased salt stress still is resulting in the sodium going into the vacuole and then accumulating there. But in this case, for the mutant, which at the level of stress which is toxic to the plant, you start to see a very different picture. You start to see that the cells are really overloaded with the sodium, and most interestingly, you can start to see that the potassium levels drop within the same within the tissue. And this is really something, again, which shows how we can look at more than one ion in parallel, and you can start to understand a consequence of stress from one ion. How does this actually reflect in the redistribution of other physiologically important ions within the plant? And so this is, I think, really I've just shown you 2 ions for today, for example, but you can see you have many different ions that sit within the cell. And so you can really start to understand what is the consequence of some of these biological processes. And in this case, how is salt stress actually reflected at the subcellular level? And so once you have these pictures, the next thing is to kind of extract some data out of this. And so in my case, I'm interested to understand the interplay between what's happening in the vacuole and the cytosol, which I've highlighted in yellow and lilac. And so basically, you can use the SEM image as your reference, because these really show you the different subcellular structures with best detail, and use this to kind of draw regions of interest onto your cryo-nanosims image and extract the count rate from these subcellular organelles. And then we can do this for each cell and for each organelle that we are interested in, and start to look at ratios of 2 different ions that we are interested to study. So in this case, for example, coming back to the ratio of sodium and potassium, for example, we start to see that we looked at the ratio of sodium and potassium in the vacuoles, which are the structures which were changing the most. And we can see that this is nicely reflected in the count rate ratios of sodium by potassium. So you can see that the ratio of sodium by potassium is higher in the wild type, which is driven by the increased accumulation of sodium in the vacuole, and this is not the case in the mutant. And you can see the opposite in the mutant. In the cytosol, where you can kind of see now that there is an increased accumulation of sodium in the cytosol, which then leads to the increased ratio of sodium to potassium in this organelle. So we're really comparing these 2 organelles, comparing them at the organelle level to start understanding what's happening within a cell. So with this, I would like to summarize and say that I hope I have shown you a good overview of how cryo-nanosims is really very useful for biology now, and it's really revolutionizing the way we can actually answer questions in biology, and with its high resolution and subcellular level understanding that we can get from this. And I've given you an overview of the cryogenic workflow, and that is required to set in place to actually be able to do this. And I've shown you one example of its potential for plant biology, but as you can see, this is just the start, and we've just answered one fundamental question, but I think there's an immense amount of work that can be done with this. And yeah, so this is just the start of a big revolution, I would call it. But yeah, and so There are many people who need to be thanked because it's not simple getting a big workflow like this in place. And so, of course, I would like to thank Anders Meibom for giving me the chance to use this fantastic instrument that he's built with his team. And also, of course, I call them the cryo crew. So this is Christina, Florent, Stefan, who I actively work with on a day-to-day basis in the different steps of the workflow. And I would like to also thank the LGB team members who have had so much fun working with. And I get very different exposure with them on different topics other than plants, for example. And also Christelle for helping with her advice and getting through the different steps of the cryogenic workflow. And of course, Nico, who I cannot not thank for his expertise in biology and giving me exposure, keeping me still tethered to the world of plants, so I don't get fully lost in the technology. And of course, the Galno Lab members, and especially Etienne also, who's been helping to follow up some of the interesting questions that have been raised from the data that we get from the cryo-nanosims, and the DBMV department also, where I very often pop up. So thank you, and I'm happy to take some questions.